Today, we're tackling a topic that I think fundamentally changes how we picture the most basic unit of life, the cell. If you took biology in high school or, even in college a few years ago, you probably have a very specific image in your head of the cell membrane.
Oh, the famous fluid mosaic model, right?
Exactly. The idea that the cell membrane is this dynamic, shifting sea of lipids where proteins and molecules just sort of float around randomly. It's a great image. Driftwood floating in an ocean, bumping into things by chance. But looking at the research we're covering today, that model feels a bit, incomplete. It implies chaos.
It does. It really implies chaos. And for a long time, that's exactly how we taught it. But the reality is much more intentional. The membrane isn't just a passive barrier or some random soup. It's actually highly organized. It's dotted with these specific rigid structures that act as decision-making hubs.
The source material calls them command centers, which I think is just a perfect rebrand. We're talking about lipid rafts. And the mission today is to understand how these rafts are built, how they traffic signals from the outside world to the nucleus, and this is the part that really got me. How these sophisticated command centers are hijacked by viruses to get inside us.
It's a classic case of biology's greatest strength also being its greatest weakness. These structures are essential for organizing cellular life. But that very organization creates a predictable lock that pathogens can pick.
Okay, so let's start with the architecture then. If we're moving away from the random C idea, what are we replacing it with? The lecture focuses heavily on this concept of microdomains.
Right, we call them lipid rafts, but the technical term really gives us the blueprint. GEMs, that stands for glycosphingolipid enriched microdomains.
I like the acronym GEM because it suggests something, solid and valuable amidst the rest of the messy membrane. But let's break down the chemistry because that enriched part is doing a lot of heavy lifting.
It really is.
These aren't just your standard fats.
No, not at all. They're chemically distinct. In a standard membrane, you have a lot of phospholipids just moving fluidly. But in a GEM, you have a high concentration of very specific lipids, things like globosides, gangliosides, and sulfatides.
And structurally, these are beastly molecules compared to the standard stuff. They all share this ceramide backbone sphingosine plus a fatty acid, but then they have these massive carbohydrate chains attached.
Exactly. And that carbohydrate chain is crucial for the visualization. If you were looking at this, say, through an electron microscope, you'd see these rafts sticking out. The source describes it as a
carbohydrate crown.
A carbohydrate crown. Yeah, a crown facing the outside world.
Right.
But the thing that makes the raft a rat, what makes it float together as a solid unit, is the glue. And that glue is cholesterol.
This is where the biochemistry gets really elegant. We usually think of cholesterol as a health risk, something clogging up arteries. But here, it's an architectural necessity. The lecture points out that cholesterol isn't just jammed in there. It's intercalated with extreme precision.
It is. It's all about the molecular alignment. Cholesterol has a rigid steroid nucleus, that's the bulky ring structure, and a hydroxyl group, an OH group. Now that hydroxyl group is hydrophilic. It likes water.
So it orients itself toward the surface, right along with those carbohydrate crowns we just mentioned.
Precisely. The sticky, water-loving parts all align at the surface. Meanwhile, the rigid steroid ring of the cholesterol packs tightly against the fatty acid tails of the sphingolipids deep inside the membrane.
It's like molecular Tetris. The cholesterol wedges itself in so tightly that it essentially freezes the surrounding lipids.
That's the perfect way to visualize it. It creates a phase separation. While the rest of the fluid mosaic membrane is moving around loosely, these GEMs become solid ordered platforms. They are liquid-ordered phases floating in a liquid-disordered sea.
And you need that stability. You can't build a command center on a swamp. You need a foundation. If these rafts were as fluid as the rest of the membrane, they couldn't hold the heavy signaling machinery in place.
Exactly. The rigidity allows them to cluster specific proteins together. But there is a second type of raft the lecture distinguishes, which brings us to a different kind of architecture. The GPI-anchored proteins.
This is the leash mechanism.
Essentially. GPI stand for glycosylphosphatidylinositol. It's a mouthful, I know, but just think of it as a complex tether. It combines a lipid tail that sits in the membrane with a sugar chain that holds onto a protein.
So instead of the protein just floating freely, it's physically anchored to this raft.
Yes. And this brings up a massive logistical question that I think is often overlooked in basic biology. We see the diagram of the finished cell with all these parts in place. But how did they get there? The cell is a factory. These rafts don't just appear. They have to be manufactured and shipped.
The origin story. I found this part fascinating. Because it traces the journey from the very center of the cell all the way out to the surface. It really emphasizes that the cell is a logistics company.
It starts in the endoplasmic reticulum, or the ER, that's the manufacturing floor. The protein is synthesized, and right there, immediately, it's attached to that GPI anchor.
So the tether is welded on right at the start of the assembly line.
Correct. And this is also where the glycosylation starts, the adding of those sugar chains. Then it gets packaged into a vesicle, a little shipping container, and it's sent to the Golgi complex.
The Golgi is essentially the finishing school, or maybe the detailing shop.
That's a great analogy.
Oh.
In the Golgi, the carbohydrate crowns are modified, they're polished. The cell adds more sugars, refining the structure. But here is the critical detail regarding orientation. All of this is happening inside the lumen, the hollow interior of the ER and Golgi.
And this leads to a bit of topological gymnastics, right? Because when that vesicle leaves the Golgi and travels to the cell surface,
it undergoes exocytosis. The vesicle fuses with the membrane and opens up. And because of the geometry of that fusion, whatever was facing the inside of the vesicle ended up facing the outside of the cell.
It's like turning a sock inside out.
Exactly. And that's why these lipid rafts and their carbohydrate crowns are exclusively found on the outer leaflet of the membrane. The manufacturing process guarantees they face the world.
Which is vital because that's where the signals are coming from. A command center hiding inside the cell wouldn't be much use. It needs to be on the front lines.
And being on the front lines allows them to do something remarkably dynamic. So we've established that these rafts are rigid platforms, but they aren't static. They can change shape to trap signals.
Okay, this brings us to the concept of caviole.
Latin for little caves.
It's a great visual. So we have this flat, rigid raft, but under certain conditions, proteins within the raft cross-link. They grab onto each other.
And that cross-linking creates tension. It forces the membrane to curve inward, forming a flask-shaped invagination. There's a specific protein called caveolin, it's usually shown as blue in the diagrams, that acts as the structural coat, forcing this curvature.
So the raft turns into a pouch or a flask. Why? Why do that? What is the strategic advantage of pulling the membrane into a cave?
It's a trap. By creating this small confined space, the cell concentrates the signaling molecules. Instead of having receptors spread out over a giant football field, you drag everyone into a small conference room.
So it amplifies the conversation.
Drastically. It facilitates signal transduction. A prime example here is the insulin receptor,
Which is obviously huge for metabolic health.
Absolutely. For insulin signaling to work efficiently, the receptors need to be clustered. The caveoli provide that clustering. It ensures the signal fires loud and clear. But, and this is the nuance, it can also do the opposite.
The lecture mentioned ENOS here, endothelial nitric oxide synthase. This is the enzyme that's responsible for relaxing blood vessels, right?
Correct. When ENOS is active, it produces nitric oxide, and your vessels dilate. But you can't have that running permanently. You need an off switch.
So the caveole acts as the off switch.
It acts as a containment zone. After ENOS is activated, it gets trapped in the caveoli to be downregulated. It essentially puts the protein in timeout to dampen the signal.
That is just incredibly sophisticated. The same structure, this flask shape, is used to amplify one signal, like insulin, and then dampen another, like ENOS. It's total control over the information flow.
That's why we call them command centers. They aren't just receiving mail, they are sorting it, reading it, and deciding whether to shout it to the nucleus or just shred it.
But this level of organization, it comes with a price. And the source material takes a bit of a turn here. We've built this perfect system. High concentrations of receptors, rigid platforms, specific markers like those carbohydrate crowns. To a biologist, that's beautiful engineering. To a virus, it's target.
The dark side of lipid rafts.
Right, because if I'm a pathogen trying to infect a cell, I don't want to just crash through the wall. I want to find the door. And the lipid raft is the VIP entrance.
It is. The lecture lists a whole rogues gallery of pathogens, viruses like SV40, bacteria, even prions that have evolved to exploit these structures.
So how exactly are they doing it? Is it just about latching onto those sugars?
Well, that's the first step. They recognize the specific carbohydrate crowns on the gangliosides, those complex lipids we discussed, and they use them as docking stations. But it goes way beyond just sticking to the surface.
Right, because the lecture mentions they hijack the signaling too. So they aren't just breaking in, they're messing with the wiring.
Exactly. Remember, these rafts are designed to transmit signals to the cell interior. Pathogens can modulate these signals. They can trick the cell into preventing apoptosis or cell death, or they can suppress the immune response.
So they use the command center to tell the cell, hey, everything is fine, don't worry about the invader, while they're setting up shop.
Or they use the caveolae mechanism, that little cave internalization, to get a free ride into the cytoplasm. They trigger the raft to fold inward, and the cell unknowingly swallows the Trojan horse.
It's terrifying, but it also explains why this specific area of biochemistry is so hot right now. We aren't just studying fats for the sake of knowing what a membrane looks like. If we want to stop these diseases, we have to understand the entry mechanism.
Exactly. You can't design a better security system if you don't understand the lock they're picking. And in this case, the lock is the lipid raft.
It really reframes the whole picture of the cell for me. We start with this idea of the fluid mosaic, which sounds passive and random, but we end up with a surface that is highly militarized. It has fortified islands, the gyms. It has precise manufacturing lines from the ER to the Golgi. It has active communication dishes, the caveolae, and it has constant security breaches from pathogens.
It transforms the membrane from a simple skin into really the most dynamic interface in biology. It's where the cell meets the universe.
And where the cell decides whether to let the universe in or, well, keep it out, ideally.
I want to circle back to one detail before we close out, because I think it ties everything together. The carbohydrate crown. We talked about it as a structural feature, but it really is the identity of the cell, isn't it?
It is. We call it the glycocalyx in a broader sense. Those sugar patterns on the lipid rafts are like a fingerprint. They tell other cells, I belong here, or I am this type of tissue.
And that fingerprint is held in place by that cholesterol glue we talked about. Without that rigid, hated molecule, the fingerprint would just smear away in the fluid ocean.
The structure enables the identity. Without the rigidity of the raft, the complex communication of life, from insulin regulating your sugar to your immune system recognizing a threat, would essentially just dissolve into noise.
It's a great reminder that in biology, nothing is random. Even a tiny patch of fat has an architecture and a purpose.
And usually a few billion years of R&D behind it.
Well, I'm certainly never going to look at a diagram of a cell the same way again. It's not just a circle with a dot in the middle. It's a command center. Thanks for helping us unpack the blueprints today. My pleasure.
And thank you for listening to the deep dive. If you enjoyed this look into the microscopic world, let us know. We'll be back next time with another stack of papers and another layer of reality to peel back.